Hello students, today we will learn about culture media for animal cells. So let's begin. The selection of an appropriate growth medium for the in vitro cultivation of cells is an important and essential step. The mammalian cells of an organ in the body receive nutrients from blood circulation. For culturing these cells in vitro, it is expected that they should be provided with the components similar to those present in blood. In general, the choice of the medium mostly depends on the type of cells to be cultured and the purpose of the cell, that is growth, differentiation, production of desired products, etc. The culture media may be natural or artificial. Now let's know about natural culture media. In early years, the natural media obtained from various biological sources were used. First is body fluids, tissue extracts. First, let's know about body fluids: plasma, serum, lymph, amniotic fluid, ascitic and pleural fluids, aqueous humor from eyes, and insect hemolymph were in common use. These fluids were tested for sterility and toxicity before their utility. Now let's know about tissue extracts. Among tissue extracts, chick embryo extracts was the most commonly employed. The extracts of liver, spleen, bone marrow and leukocytes were also used as culture media. Some workers still prefer natural media for organ culture. Now let's know about artificial media. The artificial media containing partly defined components have been in use for cell culture since 1950. The minimal critical minimum the minimal criteria needed for choosing a medium for animal cell cultures now we will discuss. The medium should provide all the nutrients to the cells. Second is maintain the physiological pH around 7.0 with adequate buffering. Third point is the medium must be sterile and isotonic to the cells. The basis for the cell culture media was the balanced salt solution, which was originally used to create a physiological pH and osmolarity required to maintain cells in vitro. For promoting growth and proliferation of cells, various constituents like glucose, amino acids, vitamins, growth factors, antibiotics, etc. were added and several media developed. Addition of serum to the various media is a common practice. However, some workers in recent years have started using serum-free media. The physiochemical properties of media required for tissue culture now we will discuss. Now let's know about the physicochemical properties of culture media. The culture media is expected to possess certain physicochemical properties, that is pH, oxygen, carbon dioxide, buffering, osmolarity, viscosity, temperature, etc. to support good growth and proliferation of the cultured cells. Now let's know about pH. Most of the cells can grow at pH in range of 7.0 to 7.4. Although there are slight variations depending on the type of cells, that is cell lines, the indicator pH red, the indicator phenol red, the indicator phenol red is most commonly used for visible detection of pH of the media. Its coloration at the different pH is different. Suppose in pH 7.4 it looks red. In pH 7.0 it looks orange. At pH 6.5 it looks yellow, and at pH 7.8 it looks like purple. Now let's know about carbon dioxide, bicarbonate and buffering. Carbon dioxide in the medium is in a dissolved state, the concentration of which depends on the atmosphere, carbon dioxide tension and temperature. Carbon dioxide in the medium exists as carbonic acid, that means H2CO3, and bicarbonate, that is HCO3 minus and H plus ions. Let's know about the reaction. That reaction is carbon dioxide plus water gives it's a reverse reaction with reverse reaction of H2CO3. It gives rise to H2CO3. Again reaction occurs, so it forms H plus plus HCO3 minus. As is evident from this equation, the concentration of carbon dioxide, HCO3 minus and pH are interrelated. By increasing the atmospheric carbon dioxide, the pH will be reduced, making the medium acidic. Addition of sodium bicarbonate neutralizes bicarbonate ions. Now let's know about the reaction. NaHCO3 reverse reaction, Na plus plus HCO3 minus. In fact, commercially available media contain a recommended concentration of bicarbonate and carbon dioxide tension for the required pH. In recent years, HEPS, HEPES, that is in long hydroxyethyl piperazine 2-sulfonic acid buffer, which is more efficient than bicarbonate buffer, is being used in the culture media. However, bicarbonate buffer is preferred by most workers because of the low cost, less toxicity and nutritional benefit to the medium. This is in contrast to HEPS, which is expensive besides being toxic to the cells. The presence of pyruvate in the medium results in the increased endogenous production of carbon dioxide by the cells. This is advantageous since the dependence on the exogenous supply of carbon dioxide and HCO3 minus will be less. In such a case, the buffering can be achieved by high concentration of amino acids. Now let's know about the oxygen. A great majority of cells in vivo are dependent on the oxygen supply for aerobic respiration. This is in fact made possible by the continuous supply of oxygen to the tissue by hemoglobin. The cultured cells mostly rely on the dissolved oxygen in the medium, which may be toxic at high concentration due to the generation of free radicals. Therefore, it is absolutely necessary to supply adequate quantities of oxygen so that the cellular requirements are met, avoiding toxic effects. Some workers add free radicals scavengers, that is glutathione and mercaptoethone, to nullify the toxicity. Addition of selenium to the medium is also advocated to reduce oxygen toxicity. This is because selenium is a cofactor for the synthesis of glutathione. In general, the glycolysis occurs in cultured cells is more anaerobic when compared to in vivo cells. Since the depth of the culture medium influences the rate of oxygen diffusion, it is advisable to keep the depth of the medium in the range of 2 to 5 mm. Now let's know about temperature. In general, the optimal temperature for a given cell culture is dependent on the body temperature of the organism serving as the source of cells. Accordingly, for cells obtained from humans and warm-blooded animals, the optimal temperature is 37 degrees centigrade. In vitro cells cannot tolerate higher temperature and most of them die if the temperature goes beyond 40 degrees centigrade. It is therefore absolutely necessary to maintain a constant temperature, that is plus or minus 0.5 degrees centigrade for reproducible results. If the cells are obtained from birds, The optimal temperature is slightly higher, that is 38.5 degrees centigrade for culturing. For cold-blooded animals, poikilotherms that do not regulate their body heat, example cold water fish, the culture temperature may be in the range of 15 to 25 degrees centigrade. Besides directly influencing growth of cells, temperature also affects solubility of carbon dioxide, that is higher temperature enhances solubility. Now let's know about osmolarity. In general, the osmolarity for most of the cultured cells from different organisms is in the range of 260 to 320 mosm per kg. This is comparable to the osmolarity of human plasma, that is 290 mosm per kg. Once an osmolarity is selected for a culture medium, it should be maintained at that level with an allowance of plus or minus 10 mosm per kg. Now, when there is an addition of acids, bases and rocks to the medium, the osmolarity gets affected. The instrument osmometer is employed for measuring osmolarities in the laboratory. Now let's know about the balanced salt solutions. The balanced salt solutions are primarily composed of inorganic salts, sometimes sodium bicarbonate, glucose and Heaps buffer may also be added to BSS. Phenol red salts as a pH indicator. The important functions of balanced salt solutions. Now we will discuss. First is supply essential or inorganic ions. Second is provide the requisite pH. Third is maintain the desired osmolarity. Fourth is supply energy from glucose. In fact, balanced salt solutions form the basis for the preparation of complete media with the requisite additions. Further, BSS is also useful for a short period incubation of cells. Now let's know about the complete culture media. In early years, balanced salt solutions were supplemented with various nutrients like amino acids, vitamins, serums, etc. to promote proliferation of cells in culture. Eagle was a pioneer in media formulation. He determined during 1950 to 60 the nutrient requirements for mammalian cells cultures, and many developments in media preparation have occurred since then. There are more than a dozen media now available for different types of culture. Now let's know about the culture medias. First is EMEM, that is Eagle's minimal essential medium. Next is DMEM, that is Dolbeco modification of Eagle's medium. DMEM, Glasgow's modification of Eagle's medium. RPMI 1630 and RPMI 1640 media from Roswell Park Memorial Institute. The other important culture media are Hams F10, F12, T199 and CMRL 1060. The detailed composition of these commonly used media, namely Eagle's MEM, RPMI 1640 and Hams F12 is we will discuss. The complete media in general contains a large number of components, that is amino acids, vitamins, salts, glucose and other organic supplements, growth factors and hormones and antibiotics, and besides serums. Depending on the medium, the quality and quantity of the ingredients vary. Now let's know about the amino acids. All the essential amino acids which cannot be synthesized by the cells have to be added to the medium. In addition, even the non-essential amino acids that can be synthesized by the cells are also usually used usually added to avoid any limitation of their cellular synthesis. Among the non-essential amino acids, glutamine and glutamate are frequently added in good quantities to the media since these amino acids serve as good sources of energy and carbon. Now let's know about the vitamins. The quality and quantity of vitamins depends on the medium. For instance, Eagle's MEM contains only water-soluble vitamins, example B complex, choline and inositol. The other vitamins are obtained from the serum added. The medium M199 contains all the fat-soluble vitamins, that is A, D, E and K. Also, in general, for the media without serum, PO4 3 minus, SO4 2 minus and HCO3 minus ion are involved in the maintenance of intracellular charge. Besides serving as precursors for the production of certain important compounds, example PO4 3 minus is required for ATP synthesis. Now let's know about glucose. Majority of culture media contain glucose which serve as an important source of energy. Glucose is degraded in glycolysis to form pyruvate or lactate. These compounds on their further metabolism enter citric acid cycle and get oxidized to carbon dioxide. However, experimental evidence indicates that the contribution of glucose for the operation of citric acid cycle is very low in vitro compared to the in vivo culture. Glutamine rather than glucose supplies carbon for the operation of citric acid cycle, and for this reason the cultured cells require very high content of glutamine. Now let's know about hormones and growth factors. For media with serum, addition to hormones and growth factors is usually not required. They are frequently added to serum-free media. Now let's know about other organic supplements. Several additional organic compounds are usually added to the media to support cultures. These include certain proteins, peptides, lipids, nucleosides, and citric acid cycle intermediates. For serum-free media, supplementation with these compounds is very useful. Now let's know about antibiotics. In the early years, culture media invariably contained antibiotics. The most commonly used antibiotics were ampicillin, penicillin, gentamicin, erythromycin, kanamycin, neomycin, and tetracycline. Antibiotics were added to reduce contamination. However, with improved aseptic conditions in the present day tissue culture laboratories, the addition of antibiotics is not required. In fact, the use of antibiotics is associated with several disadvantages. Possibility of developing antibiotic resistant cells in culture. Next is may cause anti-metabolic effects and hamper proliferation. Next point is possibility of hiding several infection temporarily. Next point is may encourage poor aseptic condition. Now let's know about serum. Serum is a natural biological fluid and is rich in various components to support cell proliferation. The most common used sera are calf serum, fetal bovine serum, horse serum, and human serum. While using human serum, it must be screened for viral diseases, that is hepatitis B, HIV. Approximately 5 to 20% of serum is mostly used for supplementing several media. Now let's know about the serum constituents. First is proteins. The in vitro functions of serum proteins are not very clear. Some of them are involved in promoting cell attachments and growth. Example, futin, fibronectin proteins. Proteins increase the viscosity of the culture medium besides contributing to buffering action. Now let's know about nutrients and metabolites. Serum contains several amino acids, glucose, phospholipids, fatty acids, nucleosides, and metabolic intermediates. That is pyruvic acid, lactic acid. These constituents do contribute to some extent for the national requirements of cells. This may, however, be insignificant in complex media with well supplemented nutrients. Now let's know about growth factors. There are certain growth factors in the serum that stimulate the proliferation of cells in the culture. Now let's know about platelet derived growth factor. PDGF. First, second is fibroblast growth factor, in short FGF. Third is epidermal growth factor, that is in short EGF. Fourth is vascular endothelial growth factor, that is VEGF. Fifth is insulin like growth factor, that is IGF-1, IGF-2. In fact, almost all these growth factors are commercially available for use in the tissue culture. Now let's know about hormones. Hydrocortisone promotes cell attachment while insulin facilitates glucose uptake by cells. Growth hormone in association with somatomedins promotes cell proliferation. Now let's know about inhibitors. Serum may also contain cellular growth inhibiting factors. Majority of them are artifacts, example bacterial toxins, antibodies. The natural serum also contains a physiological growth inhibitor, namely transforming growth factor beta, that is in short TGF beta. Most of these growth inhibitory factors may be removed by heat inactivation at 56 degrees centigrade for 30 minutes. Selection of medium and serum. As already stated, there are around a dozen media for the cell cultures. The selection of a particular medium is based on the cell lines and the purpose of culturing. For instance, for chick embryo, fibroblast and HeLa cells, EMEM is used. The medium DMEM can be used for the cultivation of neurons. In fact, information on the selection of appropriate medium for the particular cell line is available. We will discuss now. The selection of serum is also based on the type of cells being cultured. Let's know about how to choose serum. First is batch to batch variation. Second is quality control. Third is efficiency to promote growth and preservation of cells. Fourth is sterility. Fifth is heat inactivation. In recent years, there is a tendency to discontinue the use of serum and switch over to more clearly defined media. Now let's know about supplementation of the medium with tissue extracts. Besides serum, the culture media can also be supplemented with certain tissue extracts and microbial culture extracts. The example are chick embryo extract, proteolytic digest of beef heart, bactopeptone, lactalbumin, hydrolysate, tryptose. The chick embryo extract was found to contain both high molecular weight and low molecular weight compounds that support growth and proliferation of cells. Now let's know about serum-free media. Addition of serum to the culture media has been an age-old practice. However, in recent years, certain serum-free media have been developed. Now let's know about serum-free media. Addition of serum to the culture media has been an old age practice. However, in recent years, certain serum-free media have been developed. It is worthwhile to know the disadvantages associated with the use of serum and the advantages and disadvantages of serum-free media. Now let's know about the disadvantages of serum-free serum in media. Variable composition. Let's know about the variable composition. There is no uniformity in the composition of the serum. It is highly variable. Source, batch, season, collection method, processing. Such differences in the composition significantly influence the cells in the culture. Now let's know about the quality control. To maintain a uniform quality of the serum, special tests have to be performed with each batch of serum before its use. Let's know about the contamination. It is rather difficult to get serum totally free from all pathogens, particularly viruses. Now let's know about presence of growth inhibitors. In general, the concentration of growth promoters in the serum is much higher than the inhibitors. But sometimes the growth inhibitors such as TGF beta may dominate and inhibit cell proliferation. Now let's know about availability and cost. There is a dependence on the cattle for the supply of serum. Hence, the availability may be restricted on several occasions for political and economical reasons. Further, cost also is another factor for discouraging the use of serum. Now let's know about downstream processing. The presence of serum in the culture medium interferes with the isolation and purification of cell culture products. For this reason, several additional steps may be required for the isolation of the desired product. Now let's know about advantages and disadvantages of serum-free media. Advantages. The limitations associated with the use of serum in the media are eliminated in the serum-free media. In addition, there are two more distinct advantages. Selection media with defined composition. Let's know about the selection of media with defined composition. The main advantage of serum-free medium is to control growth of the cells as desired with a well-defined medium. This is in contrast to the use of serum wherein the growth frequently proceeds in an uncontrolled fashion. Now let's know about regulation of differentiation. It is possible to use a factor or a set of factors to achieve differentiation of cells with the desired and specialized functions. Now let's know about disadvantages. Slow cell proliferation. Let's know about slow cell proliferation. Most of the serum-free media are not uh, are not as efficient as medium added media in the growth promotion of cells. Now let's know about need for multiple media. A large number of serum-free media need to be developed for different cell lines. This may create some practical difficulties in a laboratory simultaneously handling several cell lines. Another limitation of serum-free medium is that a given medium may not be able to support the different stages of development even, even for a given cell line. Hence, sometimes separate media may be required even for the same cell line. Now let's know about the purity of reagents. The native serum does possess some amount of protective and detoxifying machinery and that can offer a cleansing effect on the apparatus and reagents, and therefore in the absence of serum, pure grade reagents and completely sterile apparatus should be used. Now let's know about availability and cost. In general, serum-free media are costlier than the serum-added media. This is mainly due to the fact that many of the pure chemicals added to the serum-free media are themselves expensive. Further, the availability of serum-free media is also another limitation. Now let's know about development of serum-free media. While designing serum-free media, it is desirable to identify the various serum constituents. and their quantities. The most important constituents of natural serum with reference to their use in cell cultures may be categorized. First is growth regulatory factor. Example PDGF, TGF beta. Cell adhesion factors. Example vitamins. Essential nutrients, that is example vitamins, metabolites, minerals, fatty acids. Next point is hormones. Example insulin, hydrocortisone. For replacing the serum and development of serum-free media, several constituents should supplement the media. Now let's know about the highlights. First is growth factors. A large number of growth factors, nearly 100, that promote in vitro cell proliferation and differentiation have been identified. Besides the factors, let's know about the some factors, some other factors. First is erythropoietin, in short EPO. Second is eye derived growth factors, that is EDGF1 and EDGF2. Third is interleukins, in short IL1, IL2. Hepatocyte growth factor, that is in short HGF. Brain derived neurotrophic factor, that is in short BDNF. Next point is phytohemagglutinin, that is in short PHA. Lipopolysaccharide, that is LPS. The growth factors may act synergistically or additively with each other or with other factors, example hormones, prostaglandins. Almost all the growth factors are now commercially available for the preparation of serum-free media. Now hormones. Let's know about the hormones. Growth hormone, insulin and hydrocortisone are the most commonly added hormones into the serum-free media. A combination of steroid hormones, hydrocortisone, estrogen, androgen and progesterone are used in formulating serum-free media for the maintenance of mammary epithelium. Now let's know about nutrients. Addition of certain nutrients, choline, ethanol, amine, linoleic acid, iron, copper, selenium, etc. are added in most of the serum-free media. Now let's know about proteins. Bovine serum albumin is the most commonly added protein. It promotes cell survival and growth. Next point is polyamines. Putrescine is the most widely added polyamine to the serum-free media. Polyamine promote cell growth and differentiation. Next is protease inhibitors. Addition of protease inhibitors, example soybean trypsin inhibitor, is done to the serum-free media for the trypsin-mediated subcultures. Now let's know about commonly used serum-free media. Several types of serum-free media have been developed for different cell lines. Finally finished. Any questions regarding this culture media, you can ask me in the comment box and subscribe my channel for more biotechnological topics. Thank you.